Thanks for the introduction, Rich. As I said, this equestrian thing I actually did, and looking back, I cannot believe I did this. Like, standing on a galloping horse and stuff, but yeah, a long time ago. Today, let's leave the horses aside. Let's focus on the immunity for tonight. So what you're actually seeing here is an image of a human thymus slide, and I will tell you much more about this later, but I just wanted to have a little teaser in there, like this is actually data that we've generated in the lab. And I thought I'll start off with just generally talking a little bit about immunity and what we think, or what we mean when we talk about immunity and why we actually need this. So basically, a definition of immunity is the body's way of protecting ourselves against harmful non-self. By non-self, I literally mean anything that shouldn't be in our body that we don't need for functioning. This includes, for instance, parasites, viruses, certain types of bacteria. We don't want these. As soon as they breach our barriers, breach our skin, breach our mucous membranes, we will get sick from them. But then we also have some other type of bacteria here that I depicted in blue that we actually do want to have around. So we need those in our gut microbiome on our skin, and they actually help us digest food or just generally help us stay healthy by also competing with these guys that we don't want to have around. Then we constantly challenge our body by simple things like having a beer now or eating. All of these things to the body look like something foreign, like this doesn't belong to our body, but obviously we need it, we need to get the nutrients out of it. And then lastly, the other things in our body are the cells that belong to our body, like cells in the lung that can do gas exchange so we can breathe, cells in our muscles that can contract so we can move. But on a molecular level, all of these guys look very, very similar to one another. Like all of these, whether it's a peanut, an egg, a cell in the lung, or a virus, they're all built from little proteins, they're built from lipids. So how can our body distinguish what it should actually have and what it shouldn't have, and figure out what is harmful non-self? So as with the variety of all of these invaders that we might see, our immune systems have evolved to also have a large variety of cells that are specialized to tackle any of the ones that we see up here. So these are sort of our first line defense immune cells. They're called cells of the innate immune system that just hang out under all of the barrier sites, like the skin and so on. And as soon as there is a breach by, say, a bacterium, these guys get activated and call the cavalry in, which are these more specialized types, cells of the immune system. They're called the adaptive immune system. And they're adaptive because they've got these little guys sticking out on their surface. And these are receptors that are specific to a particular kind of antigen, a particular kind of pathogen, say. So we might have a cell like this that can, it's flu season now, so we will have some of these guys that are very specific to flu, and they can detect them very quickly and very efficiently make sure that the flu virus can't spread around. Whereas these guys down here, they're very good at generally keeping viruses at bay, but they're not very good at specifically making sure they get rid of the flu. There's two cells of this type. These are T cells and these are B cells. B cells you might have encountered in the context of antibodies. When people talk about we have some antibody responses, or when you get a vaccine and you come a few years later and they check your titers to see if you still have immunity, they would measure these antibodies that these B cells maybe. In my lab, we're particularly interested in these T cells. They don't make antibodies, although they have receptors that look very similar to these ones up here. But these cells are specialized to detect cells in the body that are infected with, say, a virus or a bacterium, or that are non-self because they have turned tumorous. And these cells can detect them with their specialized receptors and can induce these cells to die. with the idea that you're infected, you're just gonna, as a cell, you're just gonna be there and you're gonna spread the virus. So if I get rid of that cell, the virus can't spread anymore. Similarly, if I can get rid of the tumor cells, they can't multiply anymore. So these guys are very powerful, but that also means they can be very dangerous. What if they look at a healthy cell in our body and mistakenly think, oh, this is actually a cancer cell, or this is a cell that's infected? They will start attacking the cell, and we're losing healthy tissue. And this is the situation where we get autoimmune diseases. If your body starts, with these type of cells, starts attacking your own tissue. So we are really interested in how do these cells figure out what they should attack and what they shouldn't attack, and what the molecular clues for this are. So I'll focus on these T cells, and basically the challenge that they face is they get to interact with all the other cells in the body, and they only get to look at them briefly. And when I say look at them, I mean they use their receptor here and bind to a receptor on the other cell. And that little interaction allows them to figure out, are you a healthy cell, or are you a sick or infected or tumor cell? And this works because all the cells in our body also have these receptors sticking out here. And they constantly think of it as like a silver platter, where they constantly just show what's going on inside the cell on the outside of the cell. So when our T cells come about and look at them, they can say, okay, this little yellow dot that I see here, which is derived from the cell, is either part of the cell as it should be, or it's part of a virus. If it's part of a virus, I'll attack the cell. If it's not part of the virus, I'll leave it be. But that also means that somehow this little snippet that I get to see, the T cell has to be trained on to know whether this is self or non-self. And this is what we're working on in the lab. So basically, how does the cell know when it gets the go signal to start killing the cell? And how does the cell know that it gets a stop signal to not attack and keep the cells of our body healthy? And I thought I'll start off with something that we probably all have encountered. Anytime on the internet you register for something, this really annoying thing comes up, and it says, "Ooh, are you human? Tell me all the buses you see." If you're lucky, you only have to do it once. Oftentimes you do this 3-4 times. You're like, what is going on? But okay, we find all the buses. We click verify. As I said, if you're lucky, you're done, and it says, okay, move on to the next slide. What it also does is the companies use this to make their algorithms smarter. So it's basically using you to label images of buses and say, they make their recognition system for buses better. So they train their own algorithms. And what happens is, by doing this many times, you help them identify that this image contains a bus and this image does not contain a bus, so they can figure out they're not the same. If we do this enough times, it also helps them to say, okay, all of these images are classified as buses. If the algorithm now sees another bus, it can now realize, oh, this is similar to these, so this is also a bus. So whenever you do a CAPTCHA, yes, it's helping you to get to whatever you're logging into, but it's also like training the machine learning there. And these are the examples that are highlighted here are two very important concepts in machine learning. So the first one is discrimination, identify bus versus non-bus. And it doesn't have to be specific what non-bus is, it's not a bus. And then if I've seen enough buses, I can then confidently generalize to an image that I haven't seen and say, oh, this is also a bus. Okay, why am I telling you this? This is basically how the immune system does it as well. Okay, so we're going to play the same game. We now need to find harmless cell in this image here. Any guesses? We've got like these cells here, like hidden amongst all the pathogens. Okay, so we click verify again, and we find out that Yes, indeed, we can now discriminate that this is self and this is non-self, so we should discriminate. And we can also, if we've done this enough times, we know most of the cells in our body, and then if another cell from our body comes along, we can generalize and be like, oh, this actually also belongs to our body. We should ignore this and not start an attack. And the place where this happens, or where this, where the immune system, or particularly the T cells, learn this generalization and discrimination process, is in an organ called the thymus. There's a schematic here, and that organ sits right atop your heart. Whenever, or often when I talk about the work that we do, people are like, oh, do you mean the thyroid? I'm like, no, I know the organ we're talking about, like the thymus. So like there is two organs that sound very similar. The thyroid is here, the thymus is here. And actually it's the last organ in the human body for which we figured out the function. And until the 60s, people have actually classified it as an evolutionary accident without functional importance. And it's long been believed that it's actually serving as a graveyard for T cells. So they go there to die, but not that it's actually functioning and helping the immune system. This is actually a quote from a Nobel Prize laureate who got the Nobel Prize for realizing that there is like learned immunity. So this was a big statement happening in the 60s. And then Jack Miller came around and he was like, I do think this organ has a function. It seems crazy for evolution to keep an organ just for something to die in there. And he did some clever experiments where he realized that if he removes this organ from mice very early on when they're born, these mice would not be able to distinguish if you take an organ transplant into this mouse, basically. So they lose the ability to distinguish, is this my own organ or my own piece of skin or not. And based on that, he actually hypothesized that if you remove this, they lose this ability of this learned immunity. And he also saw that mice that did not have this organ actually all suffered from really severe infections and succumbed to this. So that was the first indication, beginning of the 60s, and then sort of late 60s towards the 70s, this idea that we actually need this organ was fairly established. And the quote I have from this is that a very careful quote, that the thymus is important in providing an adequate pool of immunologically competent cells. Back then, we didn't know that these cells are T cells or B cells, and we couldn't describe them as we can today. But those were the first indications. And I also just want to highlight, whenever we talk about somebody else's work, we usually put down there who did it and when they did it. So we can acknowledge that we're describing something that's important, but we haven't actually contributed to this. So this is work that was done in the 60s. And then from realizing this in the 60s, late 70s, it took another 30 years for people to realize how the thymus could potentially actually do this. And I'm calling it your inner mini-you because people have realized that in order for the immune system to do the little clicky bit of saying self, no self, it actually has to have the ability to see self in the thymus. But if you think a little bit about it, your muscle cells contract and they have proteins that make that happen. The cells in your eye are really good for capturing light, and with that light capture, they can see and project that back into the brain. Cells in our liver are very good at detoxifying, but the liver cells don't start contracting. If you have a photoreceptor in a muscle, it's going to be very useless because there's no light coming in. So all these tissues have very specialized proteins that they need. And it's actually quite costly for the body to make proteins. So you don't just randomly want to make proteins that are not found. So usually there's a very tight regulation of what you have, and the body only makes the things that each organ actually needs. So if now we want to do this generalization bit where we're like, okay, I've seen most of these cells, I can say this is harmless. How can a single organ do this if I need to pretend to be a muscle, pretend to be an eye? or pretend to be a liver cell. And yeah, so this was at the beginning of the 2000s where people have realized the thymus is very special in that it can sort of overcome all of these normal regulatory processes, and it does express photoreceptors despite them not being light. It does express these contractile molecules, and really all with the purpose of actually getting our T cells educated so we have a safe immune system. So as I promised you at the beginning, we're going to go back to this slide. Actually, Rishu introduced me, and Dina, who won the raffle, are the two people who have actually taken this image. So this is an image of a human thymus slide where everything that you see in blue is a cell nucleus, sort of part of the cell where the DNA is contained. And because there's so many developing T cells in the thymus, everything pretty much looks blue. And then the areas that you see in green are the cells that are, is a fluorescent marker that recognizes cells that have this ability to pretend to be any other cell in the body, or at least express proteins like any other cell in the body. And what we do in the lab is we're interested to study how these T cells that we have here, if you remember the receptor and they do the scanning, how they interact with these green cells or the cells from the green regions here to figure out this generalization task. So we look at all of these cells that we have in the thymus and look at their molecular signatures. So what is it that they're expressing that allows them to train these T cells to put these little fragments up here? But then they also look quite different. So we can try and figure out how these cells not only train the T cells, but actually give structure to the organ. Like they're also like the integral part that allows like the T cells to go out. So we study these cells in detail because they're really key for this generalization process. And as I told you, we call this our inner mini U, and it's because the resolution is maybe not good enough. This is all the tissues in the body. So somehow these cells actually manage to represent everything else that's going on in the body. So T cells, during their development or education, as we like to refer to it, actually get to see all of you by just hanging out in the thymus and do generalization. Then we're also interested in how these T cells interact with these cells, and based on these interactions, make decisions on what type of T cell they want to become. If you remember my first slide, we were talking a little bit about all the vast majority of invaders or pathogens that we have. We have parasites, we have viruses, and we all meet them with like the most powerful tools that we have, namely cells that specialize to attack a worm, or cells that specialize to attack cells that are virus infected, but they all start off in this organ. So what are these different cues that push them down this line to become specialized? And then the third branch in the lab is really interested in this tiny interaction here. So because this is what everything hinges on. These two receptors interacting with a small snippet of protein that's coming from the cell that's driving most of these decision-making processes. So what is it that a T cell actually sees, and how can we characterize this interaction, and how does it help our immune system? And I thought I'll just show you just a few sort of the techniques that we're doing, and then I'll just open up the floor for questions. So a lot of this work is actually done mostly computational, because there's a lot of data out there already. I told you the thymus the function was discovered in the 60s. How it does it, the first clues came in the 2000s, and in the last 10, 15 years, we've really seen an explosion in techniques that can generate lots and lots of data in high throughput, where we can measure everything that's happening in gene expression space here, or we can measure all the proteins that a cell can do instead of just looking at one protein at a time. So now we have all of these techniques that give us a lot of data. We can measure many things high throughput. So we can develop new computational tools that leverage this data and can try to make predictions. To see how T cells meet their fate, we're using a lot of the techniques that I just mentioned to actually generate this data and look at different types of T cells, and then combine this data from human samples, and then also look at mouse samples, because ultimately the human samples are very important for us to actually understand what's happening in human. We don't want to cure a mouse of cancer or autoimmunity. We really want to do this in human. So we need to understand what's happening in human, but at the same time to try and figure out how we can intervene. We cannot do this in human. So we need to figure out what is the right system that we can perturb, like what is important in human and what is a good reflection of this in mouse, to figure out how we can study this. So we've been doing lots of comparative work here. And this is how my lab started off, and this is what a big focus is really trying to characterize these guys, because they're, as I said here, like the key players to allow this. If you are a scientist that does do any type of genomics, these types of diagrams you'll see in like 80-90% of any sort of paper that you're currently reading. We would call this something like a UMAP of single cell data, where every little data point here is a single cell for which we've measured the entire gene expression repertoire. And then we use some computational techniques that said, okay, we cannot look at 60,000 things at once. Can we somehow extract something that's more meaningful to the human eye, and we can look at it in 2D? So this is like a representation of about 100,000 cells from a human thymus, to try and understand what are these different guys, and can we understand how what's happening inside is actually related to their morphology, and that's what's happening here. This is a microscopy image where we're just going through a 3D stack of this particular cell. This is a cell in the thymus that takes on some of these muscle properties. It looks like it can actually contract. It has the morphology of a muscle, very long, has these spindly fibers in it. So we're interested in bridging the gap between the molecular measurements that we get and the function, or the morphological measurements, with the idea that we try and infer function from it, to move a little bit beyond just description in human. And then another very exciting avenue at the moment is, can we actually make, we're calling the thymus our mini U, can we actually make mini U's in a dish? So we get human thymus samples, and we can grow them in a dish, and these little structures are called organoids, and they built these 3D structures that can resemble the different cell types that we have in the human thymus. And we're optimizing these to try and figure out, if we now put T cells in, can we recapitulate this entire process in a dish, and thereby can try and perturb it without having to actually go in the mouse model. And so all of these things together allow us to look at this process in a very multifaceted manner. And I want to highlight this because in order to do this, I get to present all of this work, and I still enjoy that I get to actually do a little bit of the science, but a lot of what I do is actually writing grants for science, writing papers. And these are all the people in the lab that actually do all of this cool work. And the reason we can do all of this is because they also come from a variety of different backgrounds, like people with degrees in biomedical engineering or biochemistry, but then also like Rishi, who you've met, who has degrees in electrical engineering, computational biology. Amitabh, who joined with physics background and really wanted to learn how to use pipettes. Toder and Young, who are experts in the lab, but really said, listen, we can generate so much data, I want to be able to analyze my own data. So we have a wide spectrum of coming from purely experimental, moving this way, or maybe being more computational, like Amitabh, and trying to move this way. And yeah, so we rely, or I get to do all of the cool science because of the people in the lab, but then also the facilities at Cold Spring Harbor that make that work. We can only do this because Cold Spring Harbor has really cool, really good infrastructure to allow us to do these high throughput experiments, to do the microscopy. I told you that we do a lot of computational work. We couldn't do this without the IT department, the computational scientific infrastructure that we have to do this type of work. And then also collaborators at Cold Spring Harbor, but science is really a joint endeavor. Like, despite me saying like we have all of these different expertises, which is true, like in order to tackle these questions in a really deep and meaningful manner, we often rely on expertise that comes from outside our lab. So we do work with people internationally or within the US to like support the types of techniques and questions that we actually can't answer in my lab or in Cold Spring Harbor. And then all of this is only possible because there is a lot of public funding, both federal funding, majorly through the NIH in my lab, and then also through some local healthcare providers here and some private foundations. And really, science only works because all of these players are together. And so I just want to end with this last slide in case there are any more questions.
Let's give a big round of applause for Hannah.